That it? Yeah? Can you hear me well in the back? Yeah? Okay, so thank you all for being here. My name is Mauro Copelli. I'm in the physics department of Federal University of Pernambuco in Recife, Brazil, northeast of Brazil. So it's a pleasure to be here and together with my friends, Gabriel, Pablo. And I understand we have 4 lectures, right? I have 4 lectures. Okay, so I would like to tell the story about brain criticality and collective neuronal phenomena in general. And so just so I have a very rough idea, please raise your hand, those who work in neuroscience or something like that. Okay. And those who are experimentalist physicists. 1, 2. Mathematicians. Ooh. Biologists. 0, 2. Ah, great. Okay. Excellent. Okay. So the rest is just physicists, I assume, right? Okay. Okay. This is very high. This is the outline of my talk, and it's so dense that it's useless, so never mind. Just let me go to the first topic here. This is like the underlying question I'll have all the time in my mind, okay? If the brain is critical, what is the phase transition? I know it's a lot of information already for a first lecture, but I'll come to that, okay? About why the brain should be critical, what criticality means, and etc. And then part 1 is today, part 2 is today, part 3 and 4 are tomorrow. And I always like to start with this broader context, especially for physicists. For neuroscientists, it's sometimes hard to go through these slides. The question is the following: do we have a theory for how the brain works? The brain as a whole. Say, I ask you to think of a blue elephant right now. Everybody can do it. How exactly do we think about a blue elephant? All we know is that it involves a lot of neurons, many neurons. It is a collective phenomenon, somehow. But we don't know exactly how. So the theory in itself is, how good is the theory? So let me compare this to something, right? Let give you a scale of a good theory. What is the scale of a good theory? Of course, physics, right? So this is theoretical physics, okay? So theoretical physics is a well cared for golf course, so to speak. You see, every detail here matters, right? You go to CERN, you take anti-protons with anti-electrons to make anti-hydrogens and make the spectroscopy and test the fundamental assumptions of the symmetries of the universe. That's still theoretical physics. It's pretty amazing after 300 years after Newton, right? Of course, there are some small dark clouds in the sky, dark matter, things we don't quite understand. But but it's still, it's a pretty impressive building. Compared to that, I would claim that theoretical neuroscience is more or less in this kind of field here. It's like a field trip, and you're hiking in the middle of some place that you don't know exactly what's going on, and eventually you cut a brush, and you come up with a golden mine or something. You discover something completely new that you didn't expect, because we don't know exactly how to navigate theoretically what's going on here. That's my take on the thing. I particularly like hiking, and I almost destroy my shoulder when I try to play golf, so I like to play here, okay? So please have that in mind, that very elementary questions in theoretical neuroscience have not been answered. So that's a context a little bit to motivate what I'm going to talk about next, which is the critical brain. Just so I'd like to acknowledge my collaborators in this part of the talk. First is Osami Kinouchi from Ribeirão Preto. my main collaborator in the things I want to talk about today, and many former students. So let me introduce you to some experimental results. These are the neuronal avalanches, which were detected in 2003 by a group at the NIH, by Dietmar Plans and John Beggs. So they take a slice of the rat cortex. So you kill the rat, you remove its brain, you slice it, and you preserve it in a Petri dish, at the bottom of which there are 60 electrodes. So it can measure spontaneous electroactivity of the slice of a rat brain. And they have 60 electrodes, and they are measuring what is called local field potential, with some kind of average electroactivity around this electrode. So it kind of each electrode here is sensing thousands of neurons around it. Okay? So they measure some kind of electric potential. So if you look here in the 60 electrodes and a long time in the scale of seconds, what they have observed is the following. You have some kind of silence, nothing happens. And then all of a sudden, boom, many of those neurons show this very high deflection. If you put some kind of threshold, you can define a binary event, okay? Something happened here in this electrode, or something didn't. And then you wait again, and you'll see again something here. That looks, at this scale, it looks synchronous, but actually it is not. So if you look at, again, this is a longer time scale of 60 seconds, and if you look at a finer scale of a couple of tens of milliseconds, what you see is that this silence, and then these deflections, these binary events in different electrodes here, and then some silence again. So at the scale of milliseconds, this is clearly not a synchronous event. You have some spatial temporal pattern that occurs in this very, very, very reduced system. It's a very artificial system. This is kind of Heisenberg-like principle of biology. The less you control your system, the more realistic it is. But then the less you control it. So the experiments are harder. So this is a trade-off, an inevitable trade-off sometimes, most of the time.
Those electrodes are really impedance. I mean, you are measuring many spikes at the same time in each electrode, or you have
here?
Or you have single neurons in each electrode?
No, you're measuring local field potentials only. This book, they're only measuring local field potentials, and then they put a threshold and with negative deflection, and they define a binary event after some threshold.
So you do not spike sort, you don't have an idea of it.
Here, they didn't do spike sorting. Then the results I'm going to show of our work, then we did spikes. Then you have to do spike sorting. So one reason why these were called neuronal avalanches is that you observe here something that occurs in real avalanches, which is separation of time scales, STS here, in the sense that the duration is much less than the recurrence interval. It's not very familiar to Brazilians in particular, but if you go a real avalanche in a real mountain with real snow, it takes a lot of time for snow to pile up before you have a relaxation phenomenon, which is very fast comparatively with the time of the accumulation process, right? So here's more or less the same, in the sense that you have some significant silence here, a well-defined silence before something happens and this thing occurs. This avalanche occurs. So operationally speaking, an avalanche is anything that happens in between two silences. Of course, in order to do that and for that to make sense, you have to define what silence is, and silence is a silent bin. And in order for that to make sense, you have to define what the size of the bin is, which is there are some tricks here, but essentially they do something very smart. They just calculate the inter-event interval across this data, and this average of this interspike interval, they use it as a bin, in the sense that, please imagine that you have like then another here, another one here. If you take a bin, which is very small, of course you have one avalanche of one event, one avalanche, another, and another one avalanche, and another one avalanche. All of them are small, right? If you take the same data, you choose the bin the size of the room, then have one single avalanche with 4 events. You collapse everything together. So there has to be some common sense, and the common sense, the rule of thumb, just to use the interspike, the inter-event interval. You let your data choose your time scale. Okay? So there's that. Then, if you come to think about it, it's a very simple system. You have 6 electrodes. The simplest possible question you could ask is the following. Okay, whenever this happens, I see that some electrodes are lighting up. How many of them light up? On average, right? Do you agree? Is that like the simplest thing you could ask? What is this? On average, how many light up? On average, plus or minus some standard deviation. That's a characteristic question one might ask. And if you make this question, if you pose this question, you find out that the question is wrong. Because what they did is collect the probability distribution of the size of an avalanche. So the size of an avalanche here is, the size of an avalanche is just the number of electrons that lead up in the avalanche. So they collect the statistics, and this is the probability distribution of an avalanche of size S. Please note that this is a log-log scale here. So however you measure your avalanches, if you use just part of your electrodes, by choosing a different density, by choosing a different number but the same density, by choosing a different definition of avalanche, just counting the peak of the local field potential, whatever. However you do it, you get this beautiful power laws whose cutoff increase with the system size here. It's almost like a simulation you do in a critical system in your computer. They obtain this experimentally. So distribution of avalanche sizes is a power law in this setup. And the exponent is minus 3/2 approximately. So that means that this is a distribution with a fat tail. Because of course, if I show this, the first thing you should ask is, so what? So what is a power law? Well, so what? Well, so this is a fat tail distribution, which means that mean size of an avalanche, typical size of an avalanche, mean plus or minus standard deviation are all meaningless questions. Think about it. If you calculate the variance of the distribution, it's infinite. So whatever the mean is, it's useless because it's not that your system has a well-defined mean plus or minus some fluctuations. It's all fluctuations all the time. So the fluctuations are not the exception. Fluctuations are the rule. They dominate the system. And if a slice of a red cortex has any significance for brain functioning, it's a very artificial system, well, then that should inform biologists how this thing is operating at some level, right? With fluctuations, with very large fluctuations. So this is what, in the jargon, you can call it non-trivial scale invariant statistics. And it's scale invariant because of this power law here. If you change the scale, it doesn't change the distribution. And scale invariant statistics is something very interesting because it's counterintuitive. Here I'm showing two pictures of Didier Sornet's books, 2 typical geological sedimentary layers, 2 pictures, okay? Please raise your hand, those who know those pictures. No? Okay, no cheating, okay? You do? You do this picture? Okay, so yeah, don't say anything, okay? Say, assume this is a sphere, okay? I cannot draw very well. I tell you this is a physical object, okay? And I ask you, what size is the sphere? Answer, I don't know, right? It could be Jupiter. It could be the hydrogen atom. There's no way. I have no idea. Now, what size is the sphere? Well, it's a couple of centimeters, right? Tens of centimeters. Now I know. Why? Because I gave you something that has a well-defined scale. So humans have a well-defined size. If you take the distribution of humans, the human heights, see? It's something, some average, plus or minus some standard deviation. And therefore I know that this is not Jupiter nor the hydrogen atom, right? Now I know. because it gave me a scale. Now, here, Didier Sornet explained the same trick with us. So if I put a knife on your neck and ask you to please tell me what is the size of a human hand in those two pictures, what would you say? Say, one finger like this. One finger is like this size, or the size of the screen, or just this tiny piece, black piece here. What is it?
How did you measure?
It's just a picture
What did you take from picture?
with a camera. Well, you see, you need more info. Why you need more information? Because you cannot tell whether this is a close-up picture of a small rock like this. It could be, right? Can you imagine? A rock this size, that you come up with a close-up and take a picture of this. It will look like this, right? Or it could be a picture from a mountain from far away as well. So you don't know, right? This is a scaling invariant distribution. This is a scaling invariant system. So in order to help us, the author then took the cap of the camera lens and put on top of the images. So now we know, okay? So this is the cap of the camera lens, and this is the cap of the camera lens. Okay? Now we know what the size of a human hand is, because we know the size of a camera lens is, right? And then he puts, look, his finger here. Now it makes sense. Right? Except that he's cheating. Look. Look. He cheated all of us. This is huge. This whole person here. He just played with the image in order to fool us. Because when I put this the previous picture here, without the person, nobody complained, right? Nobody complained that the camera lens was this size. But it's not. It's actually much smaller. Here's a real person in the same scenario. But nobody complained. Because it's a scaling invariant system. It could be anything. This is in order to get some grasp of the scaling invariant phenomena. Right? So, going back to the experiment, what Beggs and Plans did as well was measure what is called the branching ratio. So they take in some time t plus delta t, the sum activity, the number of descendant activity divided by the number of ancestor activity in the previous time being. They divide one by the other, and they do the statistics, and they obtain that something that fluctuates. This is called the branching ratio in branching processes kind of models, and it fluctuates around 1. And then this raised the eyebrows of the physicists, because when people saw this, they immediately identified something that could be related to a well-known critical process. So let me try to make this connection. This connected to a very early, much earlier literature. Well, for instance, they reproduced this result later in anesthetized rats, so it's not only a petri dish, not as lice. They also reproduced in monkeys, which were not anesthetized. I'm sorry, the rats were anesthetized. These monkeys were not anesthetized, but they were resting. They were doing nothing. And they always get the same power law distribution of avalanche size. And then, like I said, it raised the physicists' eyebrows because it connects with very early, much early results on forest fires, which are also power law distributed. Rice piles experimentally, right? Someone did experiment with this rice piles. And this is connected to Perbach's work on self-organized criticality, whose idea is very simple. Suppose I take this table, and I take a bunch of sand in the original paper. I put sand here. So there's a pile of sand which will start to grow up. I put always in the middle, okay? Then it grows up, grows up, grows up, grows up, where I'm putting the sand, right? Grows up, grows up, grows up. Eventually, it will reach some kind of critical slope, after which whatever I put will create an avalanche, which will then relax this pile, and then it will grow up, grow up, grow up, and then there's an avalanche. And then it will grow up, and there's an avalanche. And if you collect the statistics of those avalanches, it will give you a power law, and this was termed self-organized criticality, because I'm not doing anything to control it, and it goes toward this, it goes in the direction of this critical state, regardless of what I do. You see? I didn't do anything. It just self-organizedly went to this critical point. Solar flares are power law distributed. So many people have been interested in this kind of power law phenomenon for a while. And when they saw it in the brain, it reinforced this hypothesis which had been lingering around for some time. What is the critical brain hypothesis? It's that the idea, the conjecture, that the brain as a dynamical system operates near a critical point. It can be attributed to many people. Perbach and Dante Chiavo in the 90s. Some people say that Turing in the 50s. There are some reviews about this idea. So a critical point is this point, the red point here, where I'm plotting the order parameter as a function of the control parameter. And if you increase your control parameter, nothing happens to the order parameter until you reach the critical point. At which point, then it departs from 0 and grows continuously. This is the characteristic of a second-order phase transition. And here, you have a critical point. The idea is that the brain operates somewhere here. Naturally, I'll try to justify why in a minute, okay? I promise. I'll try to give you a motivation for that. If this is true, what will be the control parameters? What is the order parameters? We don't know. I'll come to that. So this is the question that I told you I'm going to have in the back of my mind all the time. If the brain is critical, I'll try to convince you why this is an interesting hypothesis. What is the phase transition? So, oops, sorry. So the prototypical example of a phase transition in physics is the ferromagnetic transition, right? In which you have some magnets in a lattice, and if temperature is low enough, the coupling, the ferromagnetic coupling, which tries to make those two spin together, beats temperature, and eventually they have some macroscopic phase. If temperature is too high, essentially they point to all directions, and on average have zero magnetization. And I'll go back to the video in a while. If you make the plot of the magnetization as a function of temperature, you have to decrease temperature enough in order to reach this critical point, at which point you have magnetization up or down. That's why you have those two branches here. And the phase transition occurs at a critical temperature Tc. And this is a movie in which, it's a very quick movie, in which we show this essentially because most of you, I'm assuming, are theoretical physicists, and like me, studied the Ising model in statistical mechanics course, and never saw the bloody thing undergoing the phase transition in real life, right? So for those who are in this situation, YouTube comes to rescue, to help you. So this is a magnet, and there's no sound in this movie. And don't do this at home. You have to be very high temperature in order to do this here. And they're going to fast forward the movie for a while. It's very hot. It's not ferromagnetic, and now it is. And again, this was very fast, right? So now this one comes with spoiler automatically because you know it's at 0 time when it's going to become. The temperature will be low enough for the system to become ferromagnetic and therefore be attracted. And then it goes up 6, 5, 4, 3, 2, 1, and there it goes. Okay? It underwent a phase transition, second-order phase transition. Okay, so is it fair to assume that the physicists more or less know what a second-order phase transition is? Yeah? Okay. I like to be interrupted, okay? Please do. Please feel free. So the brain is a dynamical system should operate near a critical point. Why on earth, right? We know that physicists have been interested in this kind of studying this point here for a while. Yeah, why? Because it's interesting. You have very interesting phenomena going on here. But what has the brain to do with it? So let me go back to just 2-slide course on phase transitions very quickly. What you expect at critical point, non-trivial scale invariant behavior near a critical point, which is typically characterized by critical exponents, well-defined critical exponents. So if you look at, I'm calling F0 the order parameter here. The way F0 departs from 0 at the critical point, sigma is the control parameter, so sigma minus sigma c to the power beta. So beta is one critical exponent, which characterizes this phase transition. If you put your system right here at the critical point, and you somehow simulate it with an external field, in the case of the magnetic system, it would be a magnetic field, right? But in other systems, it can be something else. The response to a weak, very weak magnetic field, so H is small here, is typically nonlinear. And the nonlinearity is controlled by a critical exponent, 1 over delta H. If you look at, you take 2 parts of your system, you calculate the correlation between different parts of your system which are distant from with a distance x. Typically, what you expect normally is that spatial correlations decay exponentially with some characteristic size, characteristic length. And the characteristic length psi here diverges as you approach the critical point, and therefore your correlations are no longer exponentially decaying. They have some other kinds of decay, power loss typically. And the way the correlation length diverges is also controlled by a characteristic exponent. Something else that they expect at the critical point is that if you are able to define somehow avalanches, which has some size s, then they should be power law distributed with some well-characterized exponent tau, which is precisely what I showed you experimentally, right? In the experiments by Beggs and Plans. This is what I showed you. So when they observed that, and they had this conjecture of the critical brain in mind, people immediately got excited about it. Okay, so we have some experimental evidence that this might not be a crazy idea after all. But it is a little bit crazy. Let me. Oh, I'm sorry. Before I go there, I said it was a 2-slide course on criticality. The second question is, what is universality? It's very easy to explain like this and much harder to prove in practice. Let us imagine that 2 different models. What?
If we continue, can I ask you a question?
Sure, please.
So how do you define in the framework of this problem your order parameter?
Ah, that's an excellent. So what Gabriel's question is how to define the order parameter. So I'll be able to, that's an excellent question, which I'll be able to hand wave my way around initially, and then I'll propose a specific order parameter, okay? But it's a good question. It's one of the questions, I think, in the field, okay?
Sorry, and the control parameter?
Oh, another one. You can sit by his side, yeah. Exactly. It's even worse. Yours is even worse than his, okay? Yeah. So, please keep pushing me. It's good. So, what is universality? Let us imagine 2 different models, A and B. Model A has its own set of critical exponents. The list is longer than that, okay? I'm not showing all the critical exponents. And they're not independent. They're related to each other through Wesselian relations. I'll come back to that, I promise. So I have model B with its own critical exponents. So beta, delta H, nu perp, tau, beta prime, delta H prime, nu perp prime, etc. So models A and B belong to the same universality class if their critical exponents are all the same. This is the classical definition of universality class. And it took us a couple of decades of statistical physics, a couple of Nobel Prizes to realize that essentially very few ingredients in a given model are responsible to determine those exponents. So symmetries of the model, dimensionality, blah, blah, blah. Okay? And this understanding became clear through the renormalization group. Right? So those who are familiar with it know what I'm talking about. Okay. So just because I'm going to use the concept of universality very, very roughly in the second lecture, but I'm just exposing now. Okay. and if it was like coordinate phase transitions. So why on earth would anyone assume that the brain is a dynamical system should operate in a critical point? Why would you expect the hypothesis to be true? Essentially because people start playing with brains, models of the brain in different levels, and they come to, if you look at theory or simulations or whatnot, theory predicts that scaling invariant dynamics that you obtain at a critical point provide optical transmission capacity, as shown even experimentally in the original Bags and Place paper, optimal information processing in a very specific computational task, but again, a model in neural computation, the largest repertoire of metastable states. So if you think of metastable states of some kind of model of short-term memory, this could be interesting. So he's been shown in a model as well experimentally. And optimal sensitivity and dynamic range to incoming stimuli, which is work that I published with Asami Kinouchi many years ago. And I'm going to talk about in a minute in more detail. So I'm still hand-waving my way around this. So if you look at those ideas here, you see, optimal transmission capacity, optimal information processing, large repertoire for memory storage, optimal sensitivity to incoming stimuli. These are all highly desirable for a brain, in principle, right? It looks good. Those things look things that I want in my brain, right? So the idea is that if those things are good, they are highly desirable for a brain. There should be some evolutionary pressure that will harness all the infinite ways that the biology has of homeostasis, of adaptation, to take you there somehow. So this is more or less the rationale. And there's the, I don't know, there's the even more hand-waving way of showing. I'll wait for the next slide before I do it. I'll do hand-waving at the same time that I define the order parameter, okay? To try to convince ourselves that this could not be such a crazy idea after all. So why did people get excited when they saw those experimental results of the neuronal avalanches on Dietmar Plan's lab at the NIH? Because they immediately connected those results with branching processes, which is a very simple stochastic process that people studying stochastic process courses everywhere. You can understand it analytically. And I'll try to revisit this kind of model already in the context of a brain mimic, okay? So I'm going to play with the brain model using this kind of model. So I'm going to show the example of criticality in a very simple model of the brain. Actually, this is very strong. It's a very, very simple model of a generic sensory system, not the brain exactly. It's a different part of the brain. So we know for 50 years or 60 years, how many years? Hodgkin-Huxley papers? We know for 60, 70 years, I don't know, how one neuron works. Nobel Prize, Hodgkin-Huxley, they understood how the ionic channels work. And so you have depolarization, your cell is polarized and depolarizing, and then it goes through some refractory state before it goes back to quiescence. And these are four nonlinear differential equations. It's very complicated. And we're just going to replace this by a cellular automaton with n states. So it's 0 if it's here in silence, and then it's 1 when it's spiking, and then it goes 2, 3, 4, 5, and then minus 1 when it goes to refractory stage, and then goes back to 0. So it's a very, it's an offensively simple model of a neuron, okay? But in physics, we allow ourselves to do that, right? So, and what I'm going to do, I'm going to connect those neurons in some very simple topology, an Erdős-Rényi random graph with some average connectivity here. So essentially, say I have a neuron I here, and neuron I has 4 neighbors. So the way I connect these neuron I's to each 4 neighbors is that I'm going to draw some probabilities, PI1, PI2, PI3, PI4, from some distribution of probabilities of this matrix Pij, and say this is 0.1, this is 0.9, this is 0.2, this is 0.4, 0.5, and assume for the moment that neuron I is in the red state here, and all those 4 neurons are in the 0 state. So all the 4 neurons are susceptible to spiking. They are able to spike because they are not spiking right now, right? So if neuron I spikes, then with probability 0.1, this guy is going to spike. With probability 0.9, neuron 2 is going to spike, and so on and so forth. Okay? And they are connected randomly in a mass, in a random graph. We call this sigma I the local branching ratio of neuron I, because for each spike that occurs here, 1.7 spikes are going to occur on average, right? The next time step, if those guys are susceptible. So in the limit of very weak activity, this is a good approximation. And it turns out that this model can be solved completely. So here, the control parameter for this model, not for the brain, for the model, control parameter is just the average sigma, which I can control in my network, okay? On average, how strongly, the larger the sigma, the more strongly the neurons are connected, okay? And what is my order parameter for this problem? It's just the firing rate, the average firing rate, the average density of active sites in the state 1 here. I can probably skip this equation, but I can go through it if you want. It's very simple. It's a mean field equation, which tells you the following. What is F? F is the order parameter. What is the probability that 1 neuron is spiking? What is the probability that 1 neuron is spiking? It is the following. It is the probability that it is initially quiet. It has to be quiet in order for it to spike. So first it has to be quiet. And the probability that it's quiet is that it didn't go through any of this. It went through all those states before, after firing before. And then it has to be quiet, and then it has to be stimulated. The probability that it is stimulated is the following, is 1 minus the probability that it is stimulated from some external stimulus, times 1 minus the probability that some of its neighbors are spiking, and propagate with some probability sigma over k, which is the average value of Pij to the power k. Okay, this is, if you don't get it, that's fine. I'm just trying to convince that it's a very, very, very simple equation that you can then see. In red, you have the order parameter. In blue, you have the control parameter. You can solve for the order parameter as a function of the control parameter. And what you get is this kind of plot here, which I was showing in the first place. Okay, so it's a very simple system to understand. All I have is probability connecting those neurons. So if sigma is less than 1, that means that on average, each spike I have here will generate less than 1 spike. Then, of course, what do you think will happen? Well, activity will die out, right? You have silence. So technically, in the jargon of non-equilibrium statistical physics, this is what is called an absorbing state. It goes to silence. If, on the other hand, you have a very larger than 1 sigma, for each spike that you create here, it will create more than 1 on average. And therefore, activity will increase. That's the mechanism of a nuclear bomb, right? Except that here it doesn't explode because neurons have some refractory period, so they have a maximum rate at which they can fire. So they reach some saturation effect here. They reach some finite fraction of active sites. Here, you have the critical case in which activity is just enough for neurons, for this collective activity to propagate. Okay? So this has many names in the literature. The branching process, or the Galton-Watson model of 1874. So the British were interested in understanding why some family names survive and some others don't, right? And the answer is those who has more kids win. That's essentially what we're showing here, right? And then it goes under the name of contact process by Harris, and then they all saw that those models were essentially the same. They had the same universality class, which was named direct percolation universality class. So when I say DP is direct percolation. So maybe I can now pause for a moment and go back to my tentative answer to Gabriel, I define an order parameter. But then I can go back and try to argue again in favor of this critical brain hypothesis just with my hands and your imagination, okay? Suppose this is a good model of the brain. I mean, it's a crude model, of course. It's just a hand wave model of the brain. So, and suppose we are all neurons, okay? We are all neurons. And I'm at this extreme of the auditorium, and I want to send a message to the door up there. So what do neurons do? I'll do some spikes, right? Some message. And I'm connected to some people with some connectivity K, say 10 people, with some probability P, right? Like the model. So if I try some sequence which has presumably some meaning for the brain, whatever, and we are here, assume we are here in the subcritical regime, low P, right? So whatever I do here, I can dance my way around, do crazy stuff, whatever. There, what happens? Nothing. Eventually, one spike, eventually. I do something very complicated here. There, nothing. So that doesn't look like a very efficient brain, right? It doesn't look very, I cannot pass on the message. Now, imagine the other extreme. Imagine I'm here. Here, you have self-sustained activity. So even if I don't clap my hand, you are already clapping your hand like crazy. It's a real party. All over the place. Everybody's crazy. So now I try to pass my message here. The activity there up there in the door will be crazy. Yeah, but nothing to do with what I'm sending. It's irrelevant. It's deaf to what I'm sending because it's already occupied with all the mess that's going on. So it doesn't look like a very good brain either. Here, at the critical point, sigma equals 1 means that on average, for each spike that is produced here, for one spike that's produced here, there's one spike going on somewhere else. That's the meaning of this order parameter. So if I do like 3, 4, 5, 6 spikes here, odds are that at the other side, they will have the same order, the same number of spikes, more or less, at least, which then they have some chance of conveying a message. I say 2, they will get 2, more or less. If I say 17, there'll be 17, perhaps. So you see, again, look, it's a very crude way of trying to convince you of the point, okay? But it's very convenient because the model is very simple. There are, of course, people who do what has to be done in this case if you are into this kind of thing, but which is information theory. So they calculate information theory and mutual information between two sets of variables and try to show that this thing is really optimized in the critical case. Okay? So, so this is just one example of a model in which I can define the control parameter, I can define the order parameter, and have the phase transition, and let's see what happens with it. So this is another way of showing the same phase transition. So the black curves here are sigma less than 1. This is the density of active size as a function of time. You see, there are 3 runs here, you can barely see, because they go to 0 very fast, because it's subcritical. You are here. Sigma is 0.95. You go to 0 immediately. If you are sigma 1.05, then you get this 3 different runs, always going to the self-sustained activity, and sigma equals 1. You go to 0 as well, but it takes a long time. Actually, if you measure the time it takes for you to reach this 0 here, you have a power law distributed time. You have no characteristic time for this to happen. Actually, if you, instead of taking that 10% of your initial density, you take just one site, that's the definition of an avalanche. I create an avalanche, and I wait until it's over. This is the duration of the avalanche, not only the size, is also power law distributed, with another critical exponent, which I'll revisit strongly in the last 2 lectures. So the phase transition here, to keep in mind, is one between an absorbing here and an active phase. It's a very, very simple phase transition. Very, very simple as it can get in non-equilibrium phase transition. It's death, silence, or non-death. non-silence. That's all. There's nothing more here. Yet, and for those who are this is just a particular slide for those who are complex networks aficionados, eventually if there's anyone in the audience, because the sigma equals 1 is not the most strict criterion. The correct one is to choose the largest eigenvalue of this Pij equals 1 in criticality. Okay? And in the case of a random network, they coincide with the branch duration. But if you're not a complex network aficionado, forget about it. Never mind. Now, why am I showing you this model? Because if I take this model, put it here, and impose separation of time scales by hand, which means I create an avalanche here, wait until it's over. Okay, now it's over. Now I create another one. Wait until it's over. Okay, now I create another one. Wait until it's over. So on and so forth, 10,000 times. I do the statistics of this. What do I get? I get a power law distribution of avalanche sizes whose exponent is the same as this one. It doesn't look the same because, well, the scales are different, but it's the same exponent. Exactly the same exponent. And we know this since 1874. When the branching process was first studied, we know that the exponent is precisely 3/2, 1.5. So when they discovered this experimentally, people who knew what to connect it with. It's a very well-known process. So, see, this is so nice because the exponent that appeared here connected to a model that we knew already before. The model is simple. We can treat it analytically, and it's even simple to expand with my hands, right? So it's a win-win-win-win-win situation. Like everybody's happy here today. Yeah, now. By the way, I didn't comment but the talk, the title of this first lecture is The Rise of the Retina Speculation. Okay? The title of the second talk is The Fall of the Retina Speculation. Okay? So everybody is going to work out now. Perfectly, beautifully. Okay? Until it doesn't. So neuronal avalanches are compatible with critical dynamics. And this is, you can go to, say, some reviews. This is Miguel Bunhal's reviews, in which you see all the list of critical exponents as a function of dimensionality. And you see that the exponent 3/2 is the size exponent for dimension 4, which is the upper critical dimension, which is what you expect if you have some long-range connections in any topology. So let me try to convince you in a different way why a critical brane would be good. So this is the work I did with Ozami Kinouchi. In those list of properties of critical point that I showed you, I mentioned this nonlinear response. And let me show you how this nonlinear response at a critical point can actually explain, possibly, potentially, experimental data in biology. So psychophysics is a very old field in psychology. And the reason why it's called psychophysics is that in its simplest form, it asks this kind of question. What is the human perception, therefore psycho, of the intensity, for instance, of a sensory stimulus, therefore physical? This is physics, sensory stimulus, how much light, how much sound, how much whatever. This is neuroscience, the perception of something. So psychophysics tries to connect those two axes. It's very easy to convince yourself that psychophysical responses are nonlinear. Imagine this lady here. Put yourself in her shoes. You can easily tell which one is heavier, right? 2 kilograms is heavier than 1. Obviously, there's no doubt. You put those in your hands. This guy is in some trouble. Now it's not so easy to tell which one is harder. But the difference is the same. The difference is 1 kilogram. So it's not only the difference that matters. Therefore, the response should be nonlinear. And it's actually nonlinear. People measure this in different modalities, sensory modalities. So here in brightness, the Stevens law, this is like 100 years old, more or less, tells you that the perception is proportional to the stimulus intensity to some power m, where m is called the Stevens exponent. So these experimental results, average over several subjects with some normalization, blah, 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 but it's consistently nonlinear. And they have actually usually this result. The M is usually less than 1. And I want to understand how. How do I get from a model something that has an exponent less than 1? So a bottom-up approach in the traditional statistical physics approach. So you take this table of Stephen exponents here. You see that for brightness, for smell, in several cases, it's 0.5, 0.6 here. And I'm going to claim that I can obtain these exponents from a model. And the idea is the following. Suppose that this was a 50 by 50 array of neurons, like in the model that I showed before, okay? And here I'm going to show you the stimulus intensity in the horizontal axis in log linear and in log and linear log scale, and the vertical axis, the mean activity of those networks. Let me show you what it looks like. What is weak stimulus? And no coupling between the neurons, okay? This is very boring. No coupling. So this is what weak stimulus looks like. You have some stimulus arriving, and some neurons are spiking in response to them. If you increase the stimulus, though neurons spike more often. And if you increase the stimulus even more, they spike even more often, except that they are limited by the refractory periods. So that is very boring. The more you stimulate, the more it spikes. That's simple. It's a linear response. This is a linear response. If you introduce some coupling, what happens? Remember, coupling is some probability. Now, what this will give rise to is an increase in the response. Why? Because whenever a stimulus arrives, it could create some spike, which can then propagate to its neighbors, right? So instead of one spike for each stimulus incoming, you have a couple of spikes, right? And you can increase the coupling, right? If you increase the coupling, these small waves will last longer, right? So what's going on if this is a model of a sensory system, what's going on here is that your sensitivity is increasing. Your response to a weak stimulus is increasing because your system is amplifying what's arriving. So this is good because you can get some detection here where you otherwise wouldn't if there were no connection. So connection is good, amplification is good, right? So I amplify things, I get more amplification, I get more signal. Eventually, you know what's going to happen. If I increase sigma enough, then I'll reach a critical point. And I'm choosing a specifically very weak stimulus, which will occur, because I know the simulation, at 17 milliseconds in the leftmost part of the thing here. Look. Now, you have some stimulus arriving, and then these little waves go, back and forth, back and forth. They will eventually die, but they last a long time. Another one appear here. You see, you're at the critical point at which the survival of those waves is increasing, and therefore, if you increase sigma even more, what happens? Well, you're now in a supercritical regime. This thing will never stop, because self-sustained activity is now stable. You're in the supercritical regime, right? And so this is a two-dimensional network, of course, but I can do the calculation for a random graph and give you this result here, that this is the response as a function of stimulus intensity, and precisely at the critical point here, so sigma is increasing from here to here. Precisely at the critical point, you have a critical exponent, 1 over delta H, governing your response, and it's exactly 1.5, 0.5 for this class of model, for this universality class. And therefore, it coincides with the Stephen exponents for psychophysics. And then, in physics, you are allowed to complain that my model sucks because I have this model, and it only explains the data in one point here in parameter space, right? Here, at sigma equals 1. Here it doesn't, because the exponent's 1. Here it doesn't, because the activity doesn't even go to 0. But here it does. So this is a fine-tuning. I cannot do this in physics. But I can do this in biology if I have good reason to. So if I give you a good reason why your system would prefer to be here instead of here or here, then Evolution takes care of the rest. There should be evolutionary pressure. This is, right? Oh, again, the critical exponent is predicted in the universality class, blah, blah, blah. Some experimental data around it. So this is just data from wild-type mice. This is a cross-section of the retina. And this is a rat in which, mouse, I'm sorry, not rats, in which they have knocked out the protein which connects physically with gap junction, with electrical junctions, those neurons. And therefore, this guy lacks some kind of lateral connectivity. It's like I'm decreasing my PIJs from the wild-type mouse to the knockout mouse. And what you get is that the knockout mice that have this kind of response curve in linear log scale, and the wild-type mouse have this, again, in linear log scale, this different kinds of response curves, one of which is this one, which is very unfortunate, right? Because I would like to see whether this is a power law. I would like to see whether it's a power law that I predicted. But this is a linear log scale. I don't see power laws in linear log scales. So we took this data, and I worked with the student. We did the model, and you put this in log scale. You do see a power law with an exponent 0.5, more or less, 0.6. Okay? So this seems consistent with that model. But more importantly, I'd like to go back to this and define what is called a dynamic range. Dynamic range, I'm going to convince you why your system would like to be sitting on the red curve, which is the only curve where it works, right? It's the only one where it works. Why would you be sitting here? Consider this dynamic range definition is the following. I have a response to a given stimulus and intensity. And suppose that I send you, suppose it's your nose, okay? Let's take a specific sensory system, olfaction. So this is very, very weak stimulus. So someone opens a jar of vinegar at the back of the room, and some molecules will get here and bind to my odorant receptors. So if I send you this, if I send this or this, your nose is giving you the same response, essentially, according to this plot. If I send you a lot, if I open the bottle of vinegar in your nose like this, I give you this or this, it will receive the same. So what we say is that my network cannot code for this stimulus or cannot differentiate this stimulus or this stimulus or this stimulus, because this is like a plateau. This function is hardly invertible. You see, what I send you from the physical world is a horizontal axis. What your brain receives is the vertical axis. And it has to infer what's going on outside from what it gets here. So we say that this is bad because there's saturation here and there's saturation here. So dynamic range is a very crude definition used in biology, which is the following. I take this plateau and I take this plateau. I call this interval 100%. I remove 10% here. I remove 10% here. And what is left, I make the inverse and calculate. This is the range of stimulus that my function can be reasonably inverted without any complication by noise or whatever. Okay? Again, you can do information theory if you want. We just didn't do it because biologists like this definition. It's simpler. Okay? So this is the definition of dynamic range. So look what's happening here. If my system is subcritical, my dynamic range goes, let's say, 10% here, 10% here, from here to here. Okay? Can everybody see this? Dynamic range goes from here to here. And then if I increase the coupling, what am I doing? I'm amplifying what's going on, and therefore increasing my dynamic range because these curves are going up. So this crossing here will go up here, up here, up here, up here, so that my dynamic range is going to increase. Now, as soon as the system becomes supercritical, now I have this plateau here to discount. And then the more strongly I connect my elements, the larger this thing is, the more I have to discount, and my dynamic range is going to decrease. There's a very simple analogy that I don't plan to do with you, otherwise I'm going to hurt your ears. I have a microphone. Just imagine for a moment that this was a loudspeaker. Okay? Now, if I just tap on the microphone very gently, say you can barely hear it, Now, if I slowly come closer to the loudspeaker, there will be some reverberation between the loudspeaker and the microphone, right? And then you can hear some reverb. If I tap with the same weak intensity, you can now hear it. You can hear it better because of reverberation, right? So that's a coupling. I increase the coupling, it gets better. You get here better and better, right? Keep tapping. Better, better, until what? If I come too close, what happens? Yeah, it's audio feedback in English. In Portuguese, it's microfonia. I don't know in Spanish. How you call it? Microfonia? Como se chama? Acopla. It's like perfect. Exactly what I'm saying. It's like the coupling, right? That's a coupling. Right. So then, if I keep tapping very gently on the microphone and audio feedback sets in, you cannot hear anything that I'm tapping. You only hear the audio feedback, right? So my capacity to discriminate weak stimuli is very low. Dynamic range is very low. Dynamic range is going increasing, and then bzz, audio feedback, phase transition. Now, decreases. If I approach even better, it gets worse, right? So my capacity to discriminate has a maximum somewhere in between. Where? If I make the plot of this dynamic range, this delta here, as a function of the coupling, I get a peak at the critical point, which is precisely where my theory works. Precisely here at the critical point, I have a nonlinear response which agrees with psychophysical exponents, right? And this is the good reason that presumably biology would have in order to be here. Why? Because dynamic range is maximum. And good dynamic range is good. Large dynamic range is something good. Say, from the luminosity you face in your bedroom when you're looking for a cell phone, it's very dark, to when you go to the beach in the northeast of Brazil, on a sunny day, it's like 12 orders of magnitude that you have to be able to cope with. And of course, there are several mechanisms, there are adaptation, there are whatever, but you have to cover this dynamic range. Not only in vision, in many sensory modalities. And just for the record, this is just a list of different topologies, different models, different blah, blah, blahs, whatever, to show that this idea is very robust. It's very general, the idea that dynamics range is optimal at criticality. Is it true experimentally? So I convinced Woody Shu, who was then at the same lab that I showed before, that did my lab at the NIH, to revisit their model, their experimental system, and do an experiment where they would choose one of the electrodes to stimulate and measure the response of the network to that stimulus. So what they did is the following. They have this no-drug condition in which they stimulate with different intensities. The intensities are here in red. And then they make the system more supercritical or more subcritical. How do they do this? By using drugs. So this AP5-DNQS is something that makes your network less excitable. And you see less spikes here. And you can use PTX, picrotoxin, which makes your network more excitable, and then you have more activity. So you see, here, if you collect the data as a response to the stimulus 40, 50, 90, 150, you have a response curve. The horizontal axis is a number in red, and the vertical axis is the response here. You have this for the sub-excited network, for the no-drug condition, and for the super-excited networks. You have different response curves, and you can measure dynamic range. And you can then tell me what happens. So this is the experimental prediction from the model, and this is what they obtained experimentally 3 years later. I was happy for about 1 year, because of course it's very dirty, it's biology, okay? But you do see a tendency for things to behave the same way as predicted by the model. Different types of curves here are the different animal or different cultures, and you see that the no-drug condition tends to be closer to the maximum dynamic range, confirming that what they had before, that the system tends to organize around the critical system. You have to disturb it in order for it to go supercritical here with PTX or here with this other drug. So to finish, is our interpolation a satisfactory model of neuronal avalanches? So this branching process-like models, which have as a phase formation non-absorbing versus active phases, has several pros, good things. I told you, this first lecture, everything works out. Looks how life is nice. So the neuronal avalanche exponents, tau equals 3/2 and tau t equals 2, which I didn't mention today, but I'm going to go back to later, they work perfectly. The response exponents, 1 over delta H equals 1/5, agrees with reasonably well with psychophysics and response and sensory systems. And also, this very, 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 very simple model, you all agree it's a very simple model, can predict the maximum dynamic range of criticality, which is then confirmed experimentally in a real brain slice. So people had a good reason to eventually turn this kind of model into in fact, the de facto theoretical workhorse in the field for over a decade, because it works, more or less. You see? Today, now, for this last hour, it worked beautifully. I can convince you guys. It's simple to explain several of those exponents, it makes experimental predictions, which are confirmed. So it looks nice, right? Until the next hour. Okay, questions?
I heard that there are two different types of criticalities. I think that what you described was the statistical criticality, and there is another one, dynamical criticality. Can it be? And what are the differences, and I know they are related, but I don't know how. And another question very related. When you showed all the bifurcation diagram with the phase order and the parameter order and the phase, they look very similar as the bifurcation diagrams that we saw before with Gabriel.
Thank you for mentioning that. I was going to stop them from wiping those things out because I could use it.
So
yes,
always when we have a bifurcation, we have a critical point or something? Or?
Yeah. So let me start with your first question, which is very, very hard. I wish there were only two kinds of criticality. Actually, there's a whole mess, whole zoo of propositions in that direction, which at the end of the day go back to the question I'm posing, I'm asking, which is, okay, let's assume the brain is critical as a working hypothesis. What is the phase transition? What is the order parameter? What is the control parameter? So it has to do with all that, okay? So, but going back to your second question, which is easier to answer, is that, it's here. But he was talking about, but if you have, a phase transition can only occur if you have many infinite degrees of freedom, right? But then if you go to a particular version of this model where you can make a mean field approximation, by definition, a mean field approximation is that you're reducing the complexity of this thing to some one or two numbers, which is what I did. I can write down the equation for the order parameter, just assuming this. There are many assumptions, and therefore, what you saw there, like this, this is what.
On Wednesday we're going to.
It's a trans. Can I talk about transcritical? You talk about transcritical bifurcation.
On Wednesday we're going to look at the Kuramoto model for this kind of.
Yeah, but then it's an
evolution of order parameter.
Yes, it's like a different transition though, because the Kuramoto. In this case here, it's just a transcritical bifurcation. Very simple. But see, it is a transcritical bifurcation after I took my system and made a mean field approximation. Then by definition, I reduced all the complexity of the thing to a single number, throwing away all the fluctuations, which is what the mean field approximation is. And therefore, the equations for this, the true phase transition that eventually occurs in the model, when you map it into a mean field reduction, it maps into a bifurcation. Then the phase transition becomes a bifurcation when you're mapping into a mean field model.
So this one scales linearly like the transcritical or like a square root like
linear, so beta equals 1.
Ah, okay, so it's a
Like
transcritical.
a transcritical bifurcation. So for those, beta equals 1 is very strange for physicists who are used to study phase transitions in the Ising model, right? So in the Ising model, you have magnetization as a function of temperature, and what you do is you study, well, if you're brave enough, you go study the two-dimensional Onsager solution, which is complicated, etc. But people usually, that's not where you start. You start with the mean field version of the Ising model, in which you have something like this. And then the way the magnetization departs from 0 at t minus dc. So do you remember what is exponent here? It's beta, right? It's called beta. You remember how much is beta? How much is it? How much is it? Hello? Come on. Come on. No? StatPhys next week. Come on. It's 1.5. 1.5. It's 1.5. I'm sorry. It's 1.5. 1.5 is in my mind because of the other branching process. It's 1.5. It's 0.5. And why this is 0.5? Why this is 0.5 and why this is 1? Because of the symmetries. Remember, this one has top-down symmetry. So if you remember from the deduction, you have a free energy as a function of magnetization here, which goes like this. And then when it changes, it goes like this. So it has to keep this up-down symmetry, m minus m symmetry. That's why this has to be an even function, and this is why it has to be 1.5. Whereas this kind of model that I showed, there's no up-down symmetry. 0 and 1 are not the same. 0 is everybody dead. 1 is everybody spiking. So you miss that up-down symmetry, and the exponent changes consistently. So that's an important difference. Yeah.
I am not about brains, but a static network is a good hypothesis of a brain.
Say that again?
Okay. A static, by supposing that the brain is a static network, is it a good hypothesis? Or, for example, when one measures the activity of the neurons, I think that there are a lot of sensors that are well localized, and you study the measures of that sensors and the correlation between the measures. But the brain is static, or
No.
I don't know.
The short answer is no. Nothing in biology is static, actually. And the static thing in biology, I think it's death, right? But to be more precise, I think it depends on which question you're asking specifically. So the current paradigm in neuroscience that learning, which is what we do all the time, is precisely the change in the intensity of synaptic interactions, right? So as soon as you learn something, which we do all the time, you're changing something. So your network is changing. So this is a quick way to say no, the brain is not static. But of course, depending on the scale that you're looking at, the major connection between different areas are more or less mapped, and those are more or less well-defined, or there's some, you can treat them as static depending on what your question is. So for instance, there are people who take this major connection between areas which are obtained through a different number of techniques. So how's it called? DTI, thank you. Yeah, diffusion tensor imaging, which is something it's very simple, very nice idea. Imagine you have a neuron here, okay? A neuron is a cell. It's full of water, right? So it's it has a very well-defined direction. So if you create some spin, some pulse with your fMRI machine here, and you see the spins here, they will they will diffuse much more along this direction, right? Because they're inside the neuron. So you can measure that and do this for your whole brain and then reconstruct all the tracts. They use this for measuring which net area is connected to which one. And then people take this data, for instance, to put into a connectome and then put a model on top of that and see how it works. And then in that regard, you can use the connectome as being somewhere more or less fixed, varying some intensity. I think Paulo is going to talk about this. So you have a chance of seeing an instance of where the brain is considered somehow fixed at some scale.
Thank you.
Okay?
So Mauro, when you and your colleagues talk about the brain being critical, there are many regimes where the brain is operating. When you are performing a mechanical task, you're walking, you're moving, you're singing, or you're perceiving, you reduce the dimensionality, and it's a different dynamical state than when the brain is sleeping or when you're not, perceiving something, and when your cortical activity is not concerned with the processing of some particular information. So these ideas of being critical, they are applying, or usually they are studied under which conditions? Because I assume that if you're walking through a rope, your brain is not critical because you need very precise, low-dimensional patterns so that you don't fall, right? So typically, what was the regime where you...
So this is a very good question. It's a very hard one, as usual, but that's okay. You see, it all started with slices, right? Which are doing rigorously nothing, right? And since what appeared in slices were avalanches, which is what you expect at the zero field conditions, so to speak, the zero stimulus condition, that again is very hard to reconcile with functioning brain, because a brain has to do something, and it's usually receiving some stimulus. So this, in my opinion, creates some kind of catch-22 theoretically in the field. So that's why I like our results about the dynamic range, because it's a very simple rate-based code which allows you to work away from the zero stimulus condition. And in the context of this very simple model, again, it's a very simple model, the response to a simple rate code is best at criticality. Essentially because instead of 1 over delta H is 1.5 for this class of model, essentially your response, so you have some stimulus here, some response here, it's a square root, and therefore the weak stimuli here, they're highly amplified by this infinite derivative here, right? So I'm giving just an answer on a very simple model. Of course, I know what you're asking you're asking me much more than that, in the sense that when I have to respond somehow precisely. So I think the reasoning, again, I'm going to, for the moment, just hand wave my way around it. But I think that a real animal in a real life has to face a plethora of challenges, which are somehow some unexpected, complex, and with a non-trivial statistics. And if your brain is just disorganized, like a gas, you won't have the dynamic repertoire to deal with all of that. If your brain is highly organized, like a solid, that won't work either. It's too strict. So I think the context of what you're asking me is you have to perform some motor action. Well, those motor actions can be can vary a lot, right? In principle, they should be able to vary a lot. And the rationale goes like this, that where would you find the richest repertoire of possibilities to do whatever it takes, which you don't know what it is. It will be well, so the reasoning is that in a critical system, you will have this maximal repertoire. So going from hand waving to a specific experiment to show that it's hard. It's hard. We have done it in awake rats, showing that with the data I'm going to show in the next talk, there are signs of criticality, and there are signs of stable repertoires, which change when exposed to novel experiences. But see, this is very preliminary. You want much more, but.
So, okay, let's get it here. We will have a coffee break now, and we go back in 20 minutes. Okay?
Posso fazer umas perguntas em português mesmo?
Pode, claro. Eu só preciso aprender a desligar isso aqui.
Sim.